Hello, and welcome to Kitchen Chemistry. I'm your host, Jen Chem. Have you ever wondered what gelatin really is? Why it forms a solid, jiggly mass? And what exactly is agar? We'll answer these questions today, as well as some others. Let's get cooking! So like all good scientists, I'm not just going to explain the concepts today, I'm actually going to do an experiment. So what causes gelatin to gel? So we have our control, which has no fruit in it. Our experimental, which has pineapple. This is fresh cut. And this one is cooked pineapple from a can. We're going to prepare our gelatin. So we have packs of gelatin powder. We're going to put it in the measuring cup. And then we're going to dissolve it in boiling water. We've added a cup, which is the amount that they require for the unflavored gelatin of boiling water, and we're stirring it till it dissolves. Now I'm going to pour approximately equal amounts into each of the cupcake holders so that we can see how gelatin gels differently in the presence or absence of different types of pineapple. Now, while we wait, let's pause for a moment of science. To understand gelatin, we have to understand proteins. Proteins are made up of molecules that look like this, called amino acids. They end up having both an amino end as well as an acidic end, which is why they're called that. The amino end can bind to the acidic end to allow for long chains of proteins to be made. By bonding the different ends of the amino acids, these act like paper clips where you loop them together to form longer chains. And like these paper clips, there are different side groups for the amino acids, which means they have different properties, like these paper clips have different colors. But even after you connect all of these amino acids through a reaction of the amino side with the acidic side, you don't end up just with a straight chain of amino acids like this. The protein can end up folding in on itself by having the different side groups of the different amino acids interact. These can form sheets or helixes, all kinds of different shapes. And so they become very complicated. And this is important for the protein's function. However, when you heat the protein up, some of those bonds break, so you lose some of that structure, and you end up going more towards a linear molecule. This is what happens to make gelatin. Collagen is a protein found in animals that helps provide structural support in the body. It's a triple helix structure with 3 protein chains wound around each other. However, when we heat up animal tissue that contains collagen, these weak bonds break, forming long strands. When it cools down, some of these bonds are reestablished, so the solution becomes a tangled, gooey mess. This is the property that makes it into a gel. Most commercial gelatins are made from pig skins or animal bones, which are high in collagen. It's therefore a product from an animal, which means vegetarians and vegans need a substitute. The substitute that's most commonly used is agar, which is from red algae, which is a plant, not an animal. So while we're waiting for our gelatin to set, let's do an experiment with agar. The same thing with agar. We have our control, our fresh cut pineapple, and then the cooked pineapple from a can. Like with the gelatin, we're going to add equal parts agar to each of the cupcake holders. So why did I put pineapple in my gelatin? Let's find out. We can see that the control gelled, whereas the one with the fresh cut pineapple did not. However, the one with the cooked pineapple gelled as well as the control. So what's happening here? Well, pineapple actually contains an enzyme called bromelain. An enzyme is a special type of protein that aids in a chemical reaction. Bromelain's function function is to break down protein. And what is gelatin made of? Proteins. So why did the cooked pineapple gel as well as our control? Well, the pineapple enzyme is a protein, and like other proteins, unfolds at high temperature. It unfolds so much with heat, it can't go back to its original form, so it can no longer chew up the other proteins, like gelatin. Let's see what happened with the agar. In the agar case, that each one forms a gel, and so each one ends up being solid, no matter whether or not there's pineapple in it, or the condition of the pineapple that's in it. That's because even though the agar gels, it's not because of proteins. It's actually long strands of carbohydrates. So the enzyme's not going to break down the carbohydrates, it only works on proteins. So that's it for today. I'm going to cook up some new shows and hopefully you'll keep watching. Thanks for watching and keep cooking.